I have some disclosures to begin with, first of all, notably founder and advisor to Personalis. Well, I have had a talk for a few years that was called The Dawn of Genomic Medicine, and someone said to me recently, aren't we past the dawn? Isn't it sort of morning coffee time now for genomic medicine? And the fact is that genomic medicine is actually here today. This conference has been going on for some time, and I think when we started it, maybe it wasn't here, but for rare disease, genomic medicine is absolutely here, and you may be familiar with some of the stories that are depicted here, some of the very famous ones from Nicholas Volker on the left through the Medical College of Wisconsin group, the Duke and Stanford groups from the, featured in the New Yorker story about the NGLI1 deficiency, the Beery twins. These are stories, and the last from the UDP, the Undiagnosed Diseases Program of the NIH, that have touched our hearts. We've seen them and read about them in our newspapers, but they're also not that rare anymore. These are stories that are happening every day. They're happening every day in major medical centers and smaller medical centers around the world. We're using genomic medicine, and it's here today. So, that could be the end of the talk right there. We're also even able to do things a little faster. This is a little girl, Jaslyn, who was born prematurely and suffered multiple cardiac arrests on her first day of life, whereas previously, in order to send the genetic diagnostic for long QT syndrome, which is what she was suffering from, that would have taken us 3 months and cost about $5,000. We were able, over the course of a couple of days, to get whole genome sequencing back, analyze it over 12 hours, and give the results back to the treating physicians. We really are already in a brand new world, and it's actually just very exciting to be able to talk about being in the middle of it instead of being able to talk about what's coming. But what is coming? And the fact is we haven't actually got to personalized medicine for everyone yet. It's still coming. This is one of the things that we were dreaming about when we did this first study about 5 years ago with Steve Quake. We thought, what if everybody's genome was actually in their medical record? The genomic technology could be available for everyone. At the moment, it's there for people with rare disease. It's here. It's being used every day. But it's not yet here regularly and routinely for everyone in our hospitals. So the big question is, how do we get from here to there? And I think probably more likely we're going to get from here to there through something that looks like this. I don't think it's going to be straightforward, but there are challenges, and I think when we start to face up to them, we can get there. Some of you may have seen Tommy Caldwell and his colleague climbing the dawn wall of El Capitan. I think our challenges are probably slightly easier than his challenges with this free ascent. The first problem is that the genome is complex. It's probably more complex than most of us remember because we have programs like Repeat Masker and Picard tools in order to take away some of that complexity before we have to deal with the challenge of actually aligning the genome and calling it. Large parts of the genome are repeats, first of all. We do throw away 56%. Exons are showing there at 2% of the genome. We tend to focus on the less complicated parts of the genome. We maybe forget that genes are incredibly variable in their size and in their complexity, from the smallest gene around a couple of hundred base pairs on one exon, the transfer RNA shown at the top of the graph on the right, to dystrophin, the largest gene in the genome, which is 2.5 megabases long, and only 0.6% of it is actually exonic. And I think we have to remember that the genome is complex in order to realize where the challenges are that we need to overcome. Paralogous sequence is something that keeps me up at night and gives me a cold sweat at times, because we take these short reads that are shown on the right, and most of you know this in general. Short read technology is really the clinical grade at the current time, and we align it to a reference genome. But of course, multiple reads align in multiple different places, and we have to make decisions like where does it best align? Do we align it in one place? If it aligns in two places the same, do we just align it in both places, or do we pick one randomly, or do we throw it away? And there's lots of pretty complicated decisions in that process that can have very important downstream consequences, because the constraint that is exerted on genes, for example, versus pseudogenes is very different, and one may start to vary and the other may not. And you cannot tell from a short read, for the most part, whether you're aligning a piece of DNA that came from the gene or the pseudogene. And this has caught many of us out at times when we thought we have the answer to a particular conundrum in genomic medicine and found that actually we don't. We also have to cover the genome. Covering it consistently is hard. You're probably familiar with the 56 genes that the American College of Medical Genetics had suggested should be looked at for everyone having a clinical genome test. This is a little small for you to see. But just remind you that clinical grade right now means for a clinical genetic diagnostic, unless every single base pair is called, you won't get that test back. If there are base pairs missing from an exon, that exon will not be called and will not appear in your clinical test report. We've moved from that to a world where we accept tenfold coverage at 90% or more. And that really is very far away from where we are. And that's okay. We can still make great discoveries with that. But I don't think it's acceptable for the future. I think we need to be able to do better. To give you an idea, this graph on the right, for the current technology, would basically be one line straight down the middle at 0. We would cover every base pair. And every time that graph diverges from there is a place where we're not covering to the appropriate level of coverage at the appropriate level of quality. You're probably familiar by now with the exact server that Dan MacArthur and his colleagues and others at the Broad put up. You can do this for any gene. You can do it from the browser in front of you right now. This is tens of thousands, in this case 64,000, I think, individuals. And this is a KCNH2 gene. This causes long QT syndrome. It's actually the gene that caused the syndrome of little Jaslyn that I showed you at the beginning. You can see there's a huge hole for one of the exons. It's just not there. So if we want clinical grade sequencing, we're going to have to deal with that because we can't have enormous holes. And we published a paper recently pointing this out. It was not a comfortable position for me as someone who's been evangelizing about the genome medicine for a while, but the press kind of picked up on our criticism and said, genome scans aren't ready for your doctor's office. Here's the lesson for you. Don't make off-the-cuff comments if you're talking to a journalist. I said something like, we think of genomes like an unruly teenager, and suddenly it was all over the internet. So it's a lesson. That's a lesson learned for me. It's probably not that far from the truth at the moment. I think genome sequencing has grown up, but it still has some way to go. Certain classes of variation are very challenging to call. Here's the consensus between two approaches, complete genomics and Illumina, done on the same 12 individuals. As you can see, the consensus shown in blue of the concordant calls for single nucleotide variants is pretty high. The blue on the left is at 98%, 99%, pretty good. But for indels, and indels cause more and more severe disease than single nucleotide variants do, the concordance is around 56%. And that's for all the genes. If we just look at the really important genes for disease, it's actually even lower than that. So we have some work to go in our calling of indel variation and also structural variation that's larger. Also, rare variation is the normal. And that's a challenge, because here's the site frequency spectrum for a disease I spend a lot of time thinking about, hypertrophic cardiomyopathy. It's autosomal dominant. If you look at the left, by far the largest bar are the variants we only see once. And we know this now in a way that we didn't before. So we're going to have methods of looking at variants that we have not seen before. Singletons are the norm. And what about consistency in clinical interpretation? When different groups of people look at the evidence for different variants and look at the causality for causing disease, we find they don't agree the whole time. And it takes about an hour each. So we need to do a little bit better than 70% of concordance when we try to use a simple 5-point scale. Well, so what's the answer? If these are some of the problems, then I have to give you some sense of where we are with the answer. I like this from the internet. The answer is no. Now, what's the question? We have to define community standards. And we've just heard a fantastic talk from David Hausler about one really important community standard. And I'm not going to talk more about the Global Alliance, but it's something everybody should get on board with. He also mentioned at the end the NIST group. And the NIST group actually have an outposting here now at Stanford, and we have the pleasure and privilege of working with Mark Salat and Justin Zook to help create these tools and help apply them to clinical medicine. Starting with a truth set. We don't really have a truth set for clinical benchmarking, but we need to work towards it. For example, if you want to look at the high confidence calls that came from their paper, they defined regions where we could be highly confident about the calls, and look at the known disease-causing gene variants in ClinVar, which is shown here, you find only 81% of them actually fall in those high confidence regions. So again, there's 20% of all the known disease-causing variants. These are not possibly disease-causing. These are ones we actually believe cause disease, are not in areas of the genome that we can confidently call. And that's the number we need to make much smaller. I recommend always sequencing families if you can afford it. The costs are dropping. In the early days, we did this with John West's family. We designed some tools, including this hidden Markov model inheritance state analysis, to look at what variants were shared among individuals and what variants that cause disease were being passed to different individuals within a family. Today, we're applying these sort of tools for undiagnosed disease. Here's a good example where we sequenced a quartet, all 4 of the individuals, with this difficult, severe global developmental delay. You can see the pedigree down on the right, 2 affected brothers, both with very similar findings. The family had spent months trying to find the answer, including an exome that was sent to a very reputable lab, and found no answer at all. In this case, we decided to sequence the whole quartet, and we're able, by using an inheritance state model, to narrow this to a very small number of variants and find the answer. And in fact, just as we were thinking of writing a publication about this, someone else published it in the American Journal of Human Genetics, so that saved us the bother of writing the whole paper. That's a mixed feeling if ever you have one. But at least you know that I think that we were right. And here's the magnitude of that. If you just look at the proband shown in blue, you would have 450 genes to look at and maybe 1,400 variants to overread. If you use the inheritance state model with a trio, you get down to 40 genes and about 100 variants. And if you use the quad, you get even lower down. So you're looking at something like 20 variants, which is something that's absolutely tractable. So sequencing in families is something I recommend. Something also shown by the group at UCLA recently in this JAMA paper, when they did trio analysis, it was significantly higher hit rate, higher solve rate for trios with undiagnosed disease. Well, you should get coverage. I showed you those big holes. This is from Personalis, one of the company that we'd started up partly to translate some of these improvements in quality or answers to some of the detriments in quality towards the clinical domain. And you can see in green, you can fill in a lot of these holes if you know that they're there and you pay attention to them. It's not enough just to add extra probes. You need to actually use different chemistry. And here's the RPGR gene, which causes retinitis pigmentosa, and a disease-causing variant that would be completely missed by the standard capture approaches to that. Well, global databases, you just heard about the most important one. Another one is ClinGen, which is coming along, funded by the NIH, to bring together disease-causing variants known to cause clinical disease and share really on a global scale. And I'm lucky enough to co-chair the cardiovascular group, and so we're bringing together, we hope, all the cardiovascular disease variants that are of relevance. Also, I think phenotyping, one of the important things about that quartet was if we'd known there were several other individuals around the country with the same symptoms, we could have looked at their genomes and their exomes and narrowed the search space in that way. And so that's another extremely powerful way to narrow the search space, and something that Global Alliance and these sorts of approaches, Matchmaker Exchange was another one mentioned by David, will help us do. And finally, I'd just like to mention the Undiagnosed Diseases program at the NIH has been expanded into the Undiagnosed Diseases Network with the sites that you see here, and we were happy enough, since I co-chair the group, to co-chair just in the last couple of days here at Stanford, the whole network. It's just getting going, but the idea is for the hardest conditions, for the patients and families who've traveled around the furthest and have no answers, they can be referred into this network. They can come more locally. They no longer have to travel to DC. There are groups around the country that will be able to do that and apply not just genomic technology, but really any kind of new technology to help diagnose the hardest, most unsolved cases. All right, so where are we? I think we're at the end of the beginning. We're somewhere beyond the dawn, but we're probably not at lunchtime yet. I think genomic technology has accelerated fast, and it may be even faster than any other technology in history. It's certainly striking how rapidly we've moved along. But genomic medicine is here today. It's no longer something we're talking about for the future or a couple of small case examples. It's happening on a weekly and a daily basis. But with the power of that technology comes a lot of responsibility, and we have to start to demand clinical grade quality. Prime time, I think genetic disease and non-invasive prenatal testing, you've heard about that in other forums, I didn't mention it today. Pharmacogenomics is just around the corner. You heard a nice example from Penn where they're implementing clopidogrel pharmacogenomics. That's beginning to happen. And then coming eventually, I think, for everyone is predictive analytics.